All right, so let's get back to where we stopped. We did our triglycerides, hydrogenation. You know what, real quick before we move on, I think I did not mention this prior. So this reaction here for hydrogenation. For hydrogenation, we're going to need a catalyst to do it. It doesn't just happen by adding hydrogen, is that we're going to use a nickel. We need a nickel catalyst in order to do that. So we're going to need a nickel catalyst in order to have that hydrogenation occur. So just wanted to mention that before we forget. So we won't be using palladium. Is that we'll be using nickel. We can use palladium for smaller ones, but for larger ones, for fatty acids, we'll want to go ahead and use nickel for that. All right, and then now we can get back into our proteins. All right, so proteins. Now, proteins are long, complex molecules. Now, they contain hydrogen, oxygen, carbon, uh, nitrogen, hydrogen, and every protein is made up of what we call amino acids. And there's total 20 different amino acids. So every single amino acid are going to have similar parts to them. So every amino acid has what we call an amine group, an amino group, right? And that's designated here. So every single amino acid is going to have that. Every single amino acid is going to have a carboxylic acid group. Out of our 20 different amino acids, those will always remain constant. Is that we'll have a carboxylic acid group and we'll have an amino group. And next to the carbonyl here, so next to this carbonyl, we have what we call the alpha carbon. And then next to that, this is going to be considered the alpha amine group, the alpha amine, right? So we have the alpha amine and we have the alpha carbon that are both next to this carbonyl. So what makes every single amino acid different is this R chain. And these R chains can all be different, right? Um, and depending on the group, it gives it a different type of R group. So here we notice some things here now. So now that we're looking at these, here's my carboxylic acid group. Here's my amine group. Here's my amine group. Here's my carboxylic acid group. And they're consistent in each one of these different amino acids. And what makes these amino acids different is this is my R group. This is my R group. This is my R group. And we can see that they're slightly different in each case here. This methyl is on over here. We have a methyl group beginning in the R group. This one over here includes sulfur. And this is what makes each individual amino acid different, is that we have different R groups that are attached to these amino acids. And these R groups are what make all of these amino acids into different classifications. We have nonpolar amino acids, polar acidic amino acids. We have basic and neutral amino acids. And those are just designated by the R chain that is attached to all these different amino acids that we have. So you see, and they all come with these three-letter designations here, right? And notice here, look at this. Just because you see MET, and this begins with MET, methionine, and leucine here, LEU, and it begins with LEU. but they're not always like that. This is ILE, and it begins with ISO. Right? So we don't want to always just assume that the first three letters of the amino acid is the three-letter abbreviation, because we are going to be using these three-letter abbreviations for the protein section that we're covering, and we're also going to use this for Unit 12 when we talk about nucleic acids. So here's some examples of nonpolar amino acids. Nonpolar, you're going to see in nonpolar amino acids, you're going to see a lot of these CH bonds here, these carbon-hydrogen bonds. And we can see that the R groups here just have plenty of those bonds, right? Even here, this is still, this is CH2 over here. And I want to point out a couple of these that are pretty unique here. So we've talked about chirality and molecules being chiral. And to be chiral means that we have to have four uh, groups, four things attached to this central atom, which is going to be our carbon here. So if we take a look, let me clean some of this up a little bit. I'll just clean up. Is that I'll circle the four groups. If we take a look at the alpha carbon in each one of these cases, is that here I'll circle the four different things. So we're going to have a carbonyl attached to it. We have the R group that's attached to it. We have this amine group that's attached to it. And then there's this hydrogen that's also attached to it, right? We can imagine that the hydrogen is right there. I think there was some confusion on some of these structures, but imagine the hydrogen is here and this carbon is bonded to this other carbon over here, right? So again, we have one, two, three, four different things attached to that alpha carbon there, making them chiral. But there is one amino acid that is not chiral, and that happens to be glycine. If we circle the groups around glycine, well, here is carboxylic acid, here's the amine group, here's this hydrogen, and here's this hydrogen. And both of these hydrogens are the same, making this, I'll put achiral instead of not chiral, making this glycine achiral. So it is going to be the only amino acid like that. The only amino acid that does not have a chiral alpha carbon. Now this other one over here I want to talk about is it's another one that's pretty unique here, and that is going to be proline. I'm going to draw proline slightly different here. So let me draw proline next to it. There's my alpha carbon. Here's my carboxylic acid group. So I've just kind of drawn it sideways just to get a little better angle on that so we can kind of notice this stuff here. So here is my carboxylic acid group, and this is my alpha carbon right here. So there's my alpha carbon. There's one group. There's two groups. And here we notice that proline, it is chiral when we're talking what's connected to it. So here we have a nitrogen connected. Here we have a carbon connected. So I definitely have four groups attached to this that are different. But proline is the only amino acid where the R group attaches itself. Here's my R group right here. And you see how the R group has attached itself to that alpha amine that we see here, right? And proline's the only one to do that. It's not that we don't have ring structures in them because we see phenylalanine right next to it. And we see these ring structures. We see these ring structures, but these ring structures don't wrap themselves back around and attach themselves to that alpha amino group there. Right. So proline is the only amino acid to do that, and glycine is the only amino acid that has an achiral alpha carbon. So here's some polar acidic amino acids. Now, carboxylic acid is acidic. So let me circle these two here. So here's my carboxylic acid, and this is acidic. And if we remember from organic chemistry that our amine groups are basic. So this is acidic, and this is basic. But don't forget that every single amino acid has this. Every amino acid has a carboxylic acid group. Every single amino acid has this basic amine group here. So that is not what makes an amino acid acidic or basic. In order to consider an amino acid acidic or basic, we need to take a look at the R group. And this R group has carboxylic acid. That makes this amino acid polar acidic. Is that here, again, this is what's making glutamic acid acidic, is that carboxylic acid that's on the R group. As far as basic amino acids, well, here we go, right? Here, these are bases, right? Making lysine, arginine, histidine, and again, it's not because of this amine group, because every single amino acid has that available to them. Then we have our polar neutral amino acids, and again, just different R groups that are attached to these. And I just want to point out again, right? I mean, the three-letter abbreviation that we see here, this is not GLU, it's GLN for glutamine, ASN for asparagine. ASP belongs to aspartic acid, right? GLU belongs to glutamic acid, right? So make sure you do know the difference between these. You might want to make sure that you know these three-letter abbreviations. Give me 1 second. Okay. All right. So we talked about glycine, and again, just as a reminder here, here's our R group, or these different groups here, making glycine the only one that is achiral. All right, so we've seen DNL isomers when we're talking about our monosaccharides, right? We can have, if we take a look at the second to last carbon, we look for the hydroxy group. If it's on the left, it's L isomer. If it's on the right, it's a D isomer. And amino acids have something very similar as well. So again, we would have to have a Fischer projection in order to look at these amino acids and determine if they're D or L. But we're not looking at the hydroxy group. What we are looking at is we're looking at the amine group. So if we look at the amine group, if it's on the left, you know, it's the L isomer. If it's on the right, it's the D isomer, right? But we would need a Fischer projection. So if we look at these structures up here, right, these are not Fischer projections. This is not a Fischer projection, that I can't determine if this is D or L, because all of them have this amine group on the left-hand side, but it doesn't, because it's not a proper Fischer projection. Remember, our Fischer projections are just these bare bones horizontal and vertical lines that help us determine whether it's the D or L isomer. Right. So over here, we just have this basic bare bones structure here to determine whether this is D or L. So here, right, here's my R group, here's my R group. And depending on the amine group being on the left or the right-hand side, we have our L, D, and L isomers here. Now, amino acids contain both basic groups and acidic groups. Right? That if I were to draw this simple structure here, well, that's a carboxylic acid and it's acidic. Right? Here's butanoic acid, and butanoic acid is a weak acid. If I draw an amine by itself, well, this amine by itself is a base. So this is a base, acid or weak acid, but amino acids have both groups attached to them. Both groups attached to them, and both of these groups can either donate a hydrogen or they can accept this hydrogen ion, right? It can both accept. So an amino acid has the ability to donate one or accept one. So when we're looking at these, is that here's my amino acid. Doesn't matter what the R group is. We can actually, let's turn this into glycine. So instead of saying R, we'll just say that this is CH3. But because this has an acid and a base attached to it, is that we can ionize this. We can ionize this. If this is my acid, and we know that Bronsted-Lowry acids donate hydrogens, we know that these Bronsted-Lowry bases are going to accept these hydrogens. So this hydrogen ion, right? So this is the result when we donate those. And this is called a Zwitter ion. Now, a couple of items that you want to remember when we're talking about Zwitter ions is that Zwitter ions they're made from these amino acids. And a Zwitter ion has to contain both positive charge and a negative charge on the structure. So this has a negative charge, has a positive charge, and they're both on the same molecule, that this is a Zwitter ion. If we only had one, we would not consider that to be a Zwitter ion. So here, if we only ionize the carboxylic acid, is that this is not a Zwitter ion. That this is a basic amino acid. And I know it's a base because if this is my acid, let's do a little refresher here. Here's my carboxylic acid. If this is my acid, then this must be my conjugate base. We got carboxylic acid, and then we get a carboxylate. So this is my acid, and this is my base. And if we take a look at this structure here, it looks a lot like this. So this becomes just a basic amino acid if we only ionize the carboxylic acid portion of it. That this becomes, if we only ionize the base portion of it, well, we still maintain this carboxylic acid. So this carboxylic acid, here, if this is my base, well, then this must be my conjugate acid. So this becomes an acidic amino acid. So we have so these can all potentially occur in a solution. Is that we can have a Zwitter ion, and both of the charges, we have a neutral overall charge, and that gives us a Zwitter ion. We can have an acidic amino acid, or we can have a basic amino acids. So if I put, what was that? If I put, let's say we put this amino acid here. So let's put this amino acid that is not been ionized yet. If I put this into a solution with a pH of 10, if I put this into a solution with a pH of 10, well, according to our pH scale, and actually let me make it a little bit higher here, make it more obvious. So our pH scale ranges from 0 to 14, 7 being neutral. Anything above 7 is basic, anything below 7 is acidic. So if I know that my solution is basic, I'm going to end up with a basic amino acid, an amino acid that has a carboxylate end. If I put this amino acid here into a solution that is, let's say the pH is 2. Well, that's an acidic solution, which means that when this ionizes, I'm going to have an acidic amino acid. Right? Remember that rule that we had when working with acids and bases in our buffers, right? If I add acid, I'm going to get acid. If I add base, I'm going to get more base. Right? So depending on the solution that we put this amino acid, if I put it into a neutral solution, right? If I put this into a neutral solution, I'm going to get a Zwitter ion because this Zwitter ion is neutral. So depending on the solution that we have, is the ion, the potential ion that we can make here. So we're going to make one that's negatively charged, positively charged, or just overall neutral charge. Now, these amino acids have the ability to bond with other amino acids, right? Just like we had monosaccharides. Take a monosaccharide, add it with another monosaccharide, we get a disaccharide. So here, if we take one amino acid and bond it with another amino acid, then we don't get a di amino acid. We get something we call a dipeptide. So peptides are these protein, excuse me, are these amino acids. These peptide bonds are what's formed. These peptides are formed when we bond amino acids together. And I'll point out the actual peptide bond here. So there is some, when we're creating peptide bonds, this is our peptide bond. So the peptide bond is formed between the carboxyl group of one amino acid and the amine group of another amino acid. So here, so we're going to form a peptide bond from the carboxyl group and the amine group. And then we're also going to create a little bit of water, because we do have to, we're going to end up breaking this bond here. We break this bond here, we break this bond here, and that's going to create our water. And then our peptide bond is formed, and it joins those two amino acids together. So this happens to be a dehydration reaction, is that we're removing water from these molecules, and it's a dehydration reaction. Uh, the reaction is reversible. It's that the reaction is reversible, and this is what we're going to call hydrolysis. So hydrolysis is the reverse. So if I have a peptide, which is a bunch of amino acids that are bonded together, and if I perform hydrolysis, well, I'm going to break up that protein into its individual amino acids. So here's my dipeptide, and we're going to break it up, and we'll get phenylalanine, and we'll get alanine. So that's going to be what we're going to call hydrolysis. So we have dehydration reaction to make the peptide, and a hydrolysis reaction in order to break up the peptide bonds that we have there. So these two dipeptides here, even though they contain the same amino acids, they're both made with glycine and alanine. But just because they use glycine and alanine, we can't assume that they're going to make the same exact dipeptide. That these two dipeptides are very different. If we take a look at the alpha carbon, here's my carbonyl, and here is my alpha carbon. Here's my carbonyl, and here is my alpha carbon. This particular dipeptide here, well, its alpha carbon is chiral, right? So this dipeptide is chiral because I have a methyl group on that alpha carbon, and this particular dipeptide is achiral, right? So there is a definite difference between these two dipeptides, that just because they use the same amino acids doesn't necessarily make them the same dipeptide. So we want to just make sure that we are aware of that. Now, once we make these dipeptides, and I'll actually just kind of demonstrate it here before we move on to the next one. Actually, I'll go ahead and leave that in here. Alpha is that here, when we're looking at peptides, when we're looking at proteins, is that you're going to see them the same way all the time, where you're going to see this carboxyl group on the right-hand side, and you'll see this amine group on the left-hand side. So we end up calling this one the C terminus, and this one is end up calling the N terminus. Right, so we have what's called the carboxyl or the C terminus, the amino group, and the N-terminus. And here's the reason why we have to know these three-letter abbreviations. Is that when we write out peptide chains or we write out proteins, is that we don't want to write out the whole name. There are either three-letter abbreviations or one-letter abbreviations. If you want to memorize the one-letter abbreviations, take biochem this summer, and we memorize them all the structures, and we have the one-letter abbreviation, and it's a blast, and we have a good time doing it over the summer. So here, we're going to use the three-letter abbreviation here. And we're going to assume that these, you do have to have them in there, is that we assume that those are the peptide bonds, right? Those are our peptide bonds here. So that's what happens. So we're taking the carboxyl group of this phenylalanine, and we're bonding it with the amine group of this glycine. And then we take the carboxyl group of this glycine and we make a peptide bond through dehydration and bond it to the amine group of that alanine. And then we end up getting our N-terminus and our C-terminus here. So when we look at these structures here, this is what's going to be considered a primary structure. A primary structure. And the only thing that primary structures only give us one piece of information. The one piece of information that they provide us are the sequence of amino acids in order to make that peptide chain. And that's really all the information that we're going to get from what we call a primary structure. Is we're going to get this sequence of amino acids that make up this peptide chain. So we do have other structures that we're going to look like. We're going to look at secondary, tertiary, quaternary structures. But as far as primary structures, again, we're just going to see the three-letter abbreviation. We're just going to know what amino acids we used and the order that we put them together in order to make up the peptide. Once they get longer, because these can get quite long, and once they get longer, they start to fold on one another. And we start to get some shapes from them. And this happens to be one of the shapes that we get. So this is what we consider a secondary structure. So an alpha helix is a secondary structure. And it's held together, excuse me, it's held together by these hydrogen bonds. So all of these here, all these green dashed lines here are all hydrogen bonds. So once these get lengthy, the carboxyl group, the carboxyl group of one amino acid makes a hydrogen bond to an amine group of a different amino acid. So here it is again. Here's a carboxyl group here, makes a hydrogen bond to this amine group on a different amino acid. And this is much different than a peptide. This is not a peptide bond. In order to make a peptide bond, we do what we call a dehydration reaction, and we remove the hydrogen, we remove the hydroxy group, and then we make that bond in order to put the amino acid together. These amino acids are already put together. The interaction that's occurring here are these hydrogen bonds that are occurring between that carboxyl group, between that amine group, in order to make this hydrogen bond. So this is one type of structure. This is more common than the other structure that we have, right? So the other structure that's not as common are what we're going to call beta pleated sheets. So these beta pleated sheets, you can see the pleats that are in here, like when you wear pleated pants or pleated shirt, that we see these pleats here, and they kind of form a similar structure in them. So to take another look at these, not so three-dimensional, is that we actually have two types of beta pleated sheets. One we call anti-parallel, one that we consider parallel. And I've kind of boxed these things for you so we notice here. If we take a look at the parallel beta sheet first, we notice that there's an angle here. There's a slight angle between all of these hydrogen bonds. And again, it's from the amino group of one amino acid to a carboxyl group of another amino acid. But we see that there's some slight angles, and these run what we call parallel to each other. If we follow the order, here's nitrogen, carbon, carbon, nitrogen, nitrogen, carbon, carbon, nitrogen, and they run in the same direction. They're identical, and they run in the same direction as far as the order of nitrogens, carboxyl, alpha carbons that we have there. Now, the anti-parallel run opposite. Here is carbon, carbon, nitrogen, carbon. Well, that's over here on this end, carbon, carbon, nitrogen, carbon. Here we have a nitrogen, so these are running in opposite directions. And notice that those angles are gone. All of these hydrogen bonds that are occurring between them are no longer at an angle. So there's a couple of similarities and differences between these alpha and these alpha and parallel beta pleated sheets. All right. So both of those beta pleated sheets and these alpha helixes are what we consider secondary structures. Um, Now we get what we call tertiary structures. Tertiary structures are the interaction of secondary structures. So here we have an alpha helix. Here's a beta pleated sheet. But this alpha helix is interacting with this alpha helix, and it's also interacting with this beta pleated sheet. And all of these secondary structures are interacting with each other. So we have hydrogen bonds that are occurring between these structures, right? So um, again, we have interaction of these secondary structures. So here we have a beta pleated sheet interacting with another beta pleated sheet, right? Beta pleated sheet interacting with this alpha helix. And there's attractive forces, hydrogen bonds occurring there between these secondary structures. And then we're going to get to the highest level of organization. This is what we call a quaternary structure, where we're going to get interactions of tertiary structures. So here we have an interaction of a tertiary structure, right? Is that here is a tertiary structure, here's another tertiary structure, and another one, making up what we call a quaternary structure. Quaternary structures do not have to have four tertiary structures. This one just happens to have four, right? So this is hemoglobin here, right? So it carries oxygen through our blood, drops it off, takes out CO2. But here is what we consider to be a quaternary structure, right? Is the interaction of other tertiary structures here. And then just kind of an overview that I have for if you're going to be studying later, is that here we have this primary structure, and we get this three-letter code, gives us the order, the sequence of these amino acids. Then we have what we call beta pleated sheets. Here's some alpha helixes. When those interact, we get a tertiary structure over here. And then we get quaternary structures. And you'll notice that this quaternary structure has 1, 2, 3. So there's only 3 tertiary structures here interacting, but because they're interacting, it's still going to be considered a quaternary structure. So we don't have to necessarily have to have 4 all the time. We can just have 2 even. As long as they're interacting with each other, we're going to consider that to be a quaternary structure. And we're almost done with Unit 11 here. So this is going to just a couple topics that we need to hit to make sure that we understand the differences between what we're going to call denaturation and hydrolysis, because there tends to be some kind of confusion there. So if we go back to hydrolysis just real quick, hydrolysis is when we take a protein or a peptide, right? So you can also consider this to be a small protein, right? But hydrolysis is that we're going to take this protein or this peptide here, we're going to take this and we're going to break it up. Headphones turned off. Is that we're going to break it up into its individual amino acids. So we take this peptide as protein, break all the peptide bonds up, and we separate it into its individual amino acids. Well, denaturation, thermal denaturation, is where we're going to not break up the peptide bonds, but we are going to break up the hydrogen bonds. So denaturation, so if we were going to perform hydrolysis here, hydrolysis would break off this amino acid, and it would break off this amino acid, and we'd separate it into a bunch of little separate amino acids. So what denaturation is doing, denaturation is breaking these hydrogen bonds. Breaking these hydrogen, and once we break these hydrogen bonds up, well, then we don't get the shape anymore. So this alpha helix structure is removed, and that's what occurs in denaturation. So here we have this protein that's folded, um, either with alpha helixes or beta pleated sheets, and then we start to break those hydrogen bonds, and all we get, we still have the protein that's bonded together. So all the peptide bonds are still there. It's still a long chain, but it's more just kind of like a loose ribbon now, right? So here we see alpha helixes, beta pleated sheet, but once denaturation occurs and we break all of those hydrogen bonds, that it just becomes just a loose ribbon, where there's no more shape to it, but all the individual amino acids are still bonded together in there. And that's not what happens with hydrolysis, right? So hydrolysis, when our bodies do this, when we're sitting there eating proteins, any kind of meat, any kind of protein that comes in, is that our bodies will hydrolyze that and digest it and break down those individual proteins. So here we have this whole protein, and then eventually we're just kind of breaking those down into different amino acids here. So that's what we're going to consider to be hydrolysis. So hydrolysis is going to use some enzymes in order to break up the protein and just break it up into individual amino acids. So there's no more peptide bonds in there. So this contains peptide bonds in here, but once we do hydrolysis and break them all down, that there's just no more peptide bonds here. So a large difference between hydrolysis and what we consider denaturation. So this will end Unit 11. I'm going to pause the recording real quick. All right, so this is going to be for Unit 12. This is our last unit. We'll cover nucleic acids. So nucleic acids, and it's probably a really good lead-in from the end of Unit 11, um, because we're still going to be talking about information that was relevant to our proteins. So nucleic acids hold this information for our growth and reproduction. So we have two types of nucleic acids. One we consider DNA, the other one we consider RNA. I'm sure you've probably already maybe familiar with these terms, right? Deoxyribonucleic acid, and then we have what we call ribonucleic acid. Each nucleic acid has parts to them, just like we had parts of an amino acid, the carboxyl group, the amino group, um, is that nucleic acids also have something similar. So when we look at nucleic acid, we'll break it down into three parts. Is that every nucleic acid is going to have a nitrogen base. They will all have a phosphate group, and they will all have a sugar, right? So nucleotides are going to consist of these three basic things. What makes them different is, well, we have different bases to attach to it. We have two types of sugars. Now, the type of sugar is a real giveaway for DNA and RNA. So this is how you determine the difference between DNA and RNA, and one way to determine that is the sugar that's used. So ribonucleic acid is made from what we call ribose sugar. I'll go back to these amino acids. Uh, is it not in the slide? No. Where's my slide? Did I get the wrong? Oh, no, there it is. I panicked. All right, I'm good. All right, so we have RNA and then DNA. So our ribose is the sugar that's in RNA. And we can determine that simply by just looking at carbon number 2. That carbon number 2 contains this oxygen, ribonucleic acid. And if we look at the term deoxy and we look at the same carbon, we notice that there is no oxygen bonded there, that the oxygen has been removed. So this is what we consider deoxyribose, DNA. So this is a dead giveaway if we're looking at a whole structure. If we're looking at an entire structure, is that we take a look at carbon number 2, and we can determine whether that is RNA or whether that is DNA. So if we take a look at a structure, let's see if I have a structure here. So here is a structure. If I take a look at this nucleotide here, well, I want to know if this is what we're going to call a nucleotide. And is this nucleotide for DNA or is it for RNA? Well, let's take a look at carbon number 2. And I notice that there's oxygen there, meaning that this is ribose, and this is for RNA because I see that hydroxy group there. And they don't mingle them. We don't put both of them in DNA or RNA. In RNA, this is the only sugar that's used, is ribose. It's not going to be ribose and deoxyribose, and we don't mix them up. RNA only has this sugar. DNA only has this sugar, and we'll never see them mixed together in a strand. The next thing that makes them different are what we call these nitrogen bases. So there's two types of bases, two classes, right? Um, we have two classes, pyrimidines and what we call purines. These are what we're going to call heterocyclic rings. They're heterocyclic because these rings use carbon and nitrogen. It's a six-sided ring, but there's two different elements that make up the ring, making it what we call a heterocyclic ring. So pyrimidines are just one heterocyclic ring, and purines are two heterocyclic rings. So these pyrimidines and these purines Well, other than just the two bases, or the two types, permitters and purines, we have individual ones, and these 5 individual amino acids are what make up all of our nucleotides, all of our nucleic acids. So as far as DNA concerned, we're only going to find these structures that I'm marking in red in DNA. So uracil is not found in DNA. The only 4 bases that we find in DNA are the ones that I've marked in red. So thymine, cytosine, adenine, guanine, and we just use a 1-letter abbreviation. So this is T, this is C, this is A, this is G, and we're just going to use 1-letter abbreviations for these. So these again are the only 4 that we find in DNA. If we're looking at RNA, well, these are what we're going to find in RNA. So we'll see that cytosine is found in both, adenine is found in both, guanine is found in both, but RNA does not contain thymine, right? RNA contains uracil as a replacement for thymine. And then we have our phosphate group, right? So our phosphate derived from phosphoric acid, but those 3 components are all what make up what we call a nucleotide. So know this term. You need to know what a nucleotide is, and that it contains a nitrogen base, a sugar, and a phosphate group. In order to build a nucleotide, well, we begin with just a sugar, ribose, and uh, a purine, and that's what make up what we call a nucleoside. So the nucleoside is not a finished nucleotide. A nucleoside does not contain the phosphate yet. But when we're building these nucleotides, they're first built with sugar, then base, and then we eventually add on the phosphate group. So this, and again, this is going to be for RNA, and I know that because carbon number 2 has a hydroxy group. So once the nucleoside is made, this is where we can add on the phosphate, right? So this phosphate here is added to the nucleoside, and then we get a fully formed nucleotide, right? AMP. I don't. Some of you, I just kind of mentioned this. You're not going to be tested on it, but I just kind of mentioned this during this part of the lecture. Is that for those of you that have taken biology, you already know. I'm sure some of you might have been heard the term of ADP or even ATP. So this is adenosine 5 monophosphate, AMP. ADP would mean that I have another phosphate group attached to there, and that would be ADP. And then ATP would actually even put out another phosphate group. So those phosphate groups, the difference of phosphate group make up our difference between ATP, ADP. They're all the same structure, except we have a different number of phosphate groups on there making up ADP and ATP. But again, you're not tested on that, just kind of food for thought. All right, so we have a general structure of nucleotides then, right? And what makes each nucleotide different are these individual bases. And that's what makes up the difference in these nucleotides. Uh, so this here, what makes them different is just the base. But when we chain these together, we have something we end up calling the sugar phosphate backbone, right? Sugar phosphate backbone, and then we have a base that's added to it. Sugar phosphate, we add another base, and over here we have another base. And so the bases are really what regulate the sequence here that makes each nucleic acid different, right? Because we can definitely have just a different order of these sequences in our nucleic acids. And over here, this is what we're going to call, if we take a look at this next structure, it's really complex here. So you're going to see a pretty complex structure here. And I can't turn it sideways here, but here is our phosphate. I'm going to do this in green. No, I'll do it in red here. This is our phosphate sugar backbone, right? That's what we see here. There's our phosphate sugar backbone. And then we're going to see where it says 5' and 3'. And this is always the sequence of our nucleic acid. So here, this is what we're going to call the 5', and then what we call the 3'. So that always runs 5' to 3' because that is where the phosphate groups are attached. All of these phosphate groups are attached at those carbon numbers. So carbon number 1, 2, 3. 1, 2, 3, 4, 5. So here, all of our nucleotides are bonded together between carbons number 3 and carbons number 5 of every single sugar that we have. And that's why we run this. We always state that we run this from 5' to 3'. That's that little, it's not an apostrophe there. That's a symbol that just stands for prime. So we go what we call, this is labeled 5' to 3'. And if we're looking at this sugar, well, I'm sorry, if we're looking at this nucleic acid here, there's two ways to tell whether this is for DNA or whether this is for RNA. One, we've already talked about. Well, I'm going to look at carbon number 2, and I notice that there's no hydroxy group there, which means that oxygen has been removed, that this is deoxyribonucleic acid, that this is definitely for DNA. That's one way to tell. The other way to tell that this is for DNA is that this is thymine, and thymine is not found in RNA. That is uracil. Thymine is in DNA, uracil is in RNA. And because of these structures, I mean, nobody wants to draw this out. Nobody wants to draw a bunch of sugars and phosphate groups. If it's DNA, I know it's a phosphate, and I know it's deoxyribose. If it's RNA, I know it's a phosphate, and I know it's ribose. So the phosphate and sugar kind of play an irrelevant part when we're looking at primary structures. The primary structure, what's going to make these different are the bases that are attached to these amino acids. So we could shorten this up a little bit, and we could say that we're going to go 5', and that first base is adenine. And we'll just write A. And the next base is cytosine. And the next base is guanine. And the next base is thymine, and I end it with 3'. And that really saves me a lot of time. I know just by looking at this, I don't have to have the image anymore, but just by looking at this, I can tell that it's DNA because of thymine. I already know that there's a phosphate bonded between carbons number 5 and number 3, and I know that this is deoxyribose. So there's really no need to keep repeating phosphate, deoxyribose, phosphate, deoxyribose, when all we need is just the order, the sequence of the bases. So this is really how we're going to work with nucleic acids, is just by using the primary structure here to understand them. So when we're looking at these primary structures, these bases all contain hydrogen, and they also contain, this is going to be partially negative, partially positive. Oh, let me draw that a little better. Partially negative, partially positive, partially negative. Is that here, because we have these partial charges on here, is that we're going to be able to make hydrogen bonds between these nitrogen bases. So these bond very specifically. Adenine will only bond with thymine in DNA, and cytosine will only make hydrogen bonds with guanine. Now, they're very specific. Adenine does not make hydrogen bonds with cytosine. Adenine does not make hydrogen bonds with guanine, and vice versa. Thymine and cytosine don't make hydrogen bonds. It's always A to T, C to G. Always. In RNA, we would replace thymine with uracil. So we would have A to U. Instead of A to T, we would have A to U. But again, it's going to be uracil does not bond with cytosine, does not bond with guanine. Right? Is that we either make 2 hydrogen bonds between adenine and thymine or adenine uracil, and 3 hydrogen bonds between cytosine and guanine. And they're very specific as to how that happens. Now, because, let me see if it's here. No. All right, just want to make sure I cover this. Because of this specific bonding, we can actually determine the percentage of adenine, thymine, of all these nucleotides. We know the percentage that we have because of how it matches up. That if we had, let's say, 30% of my nucleic acid was, let's see, let's say 30% of my nucleic acid was made up of cytosine. What are the other percentages? Well, it all has to add up to 100%. You don't need a calculator for this math. If I know that cytosine is 30% and it only matches up with guanine, then that also means that guanine is also 30%. This gives me 60%. Well, how much more do I need in order to determine 100? I need 40%. Well, the other 40% has to come from adenine It has to come from adenine and thymine. Well, because of this very specific nature in which they bond, well, that means that I have to have 20% adenine and 20% thymine. right? We'll do one more. Let's say we had 23% adenine. Well, what are the other percentages? All right, well, I also have to have 23. Oop, that's a horrible looking 3. I have to have 23% thymine. This comes out to 64. Nope, that comes out to not 64. That comes out to 46%. Well, I'm going to need 54% of cytosine and guanine. So cytosine and guanine, 27% cytosine, 27% guanine. 46. My math good? I think so. Yeah. But this is how we determine the percentages of each one if we're only given one of the percents, because of this very specific nature in which they do hydrogen bonding. So what happens here is all this hydrogen bonding that occurs, we don't create just one alpha helix, is we end up creating 2 alpha helixes, and this is what we're going to consider a double helix, because we see that there's 2 alpha helixes here. And here's our phosphate sugar backbone, right? Phosphate sugar backbone. And then all of the hydrogen bonding that occurs here. All of this hydrogen bonding that occurs between all of those. So this is what we're going to call a secondary structure. Our primary structure is just this, our order of the amino acids. Primary structure. Primary structure is just the order of amino acids. I'm sorry, just the order of the nucleotides where nucleic acids are. So primary structure, we're talking about proteins or amino acids, but for nucleic acids, that's the order of the nucleotides, right? Order of the nucleotides. Secondary structure is when we have all these nucleotides here, not amino acids, all these nucleotides are bonded through hydrogen bonds, and we get what we call a double helix. Just another view of it. And these do run anti-parallel. So here we could see 5' to 3', and then we go from 3' to 5'. So they do run anti-parallel to each other, right? Here's our sugar phosphate backbone. Would this be for RNA or for DNA? Well, here's thymine. I know that this is a DNA structure. So as far as writing a complementary strand, we know that we do have this complementary strand as long as we remember that A goes to T, is that C goes to G, is we can write our own anti-parallel complementary strand. So here, if I wanted a complementary strand for DNA, I'd go from 3' to 5'. I know that A goes to T, C goes to G, T goes to A, C goes to G, G goes to C, C, A, T, T. And here I would have, I should probably write that a little bit larger here. Man, my writing's not that great. I need to practice on my pad. All right, so here we have what we call a complementary strand. In that here, we know that we don't have to draw these in, but just an understanding that we do have 3 hydrogen bonds between cytosine and guanine and 2 hydrogen bonds between adenine and thymine. But you don't have to draw all those in for the complementary strand. If we wanted to do a complementary RNA strand. Well, we're not going to use thymine anymore, that we need to use uracil. So here, instead of A going to T, A would go to U. C goes to G, T goes to A, C goes to G, G goes to C, G goes to C, T goes to A goes to U, and A goes to U again. And now we have a complementary RNA strand. Right. So that's how we would do our complementary RNA or DNA strands, uh, knowing this specific pairing, complementary base pairs that happen between these bases here. So DNA, every chromosome here, right, is one molecule of DNA. So every chromosome is one molecule of DNA. Now, the DNA is wrapped around what we call these histones here, right? So all of our DNA is wrapped around these histones. And these little snippets here are just, so our chromosomes are just filled with different genes that have a specific job in mind that, you know, make this particular protein, right? These are directions to make a particular protein. These are directions to build a certain type of cell, right? And all of that is involved in our DNA. Um, and we find all of these chromosomes. So there's 46 chromosomes, each containing one molecule of DNA, all found in the nucleus. And you know, it amazes me. I'm always just amazed by this. I mean, everything about me is all broken down to some basic chemistry. Some basic hydrogen bonding that occurs here is what dictates everything about me. The color of my skin, the color of my eyes, the way I talk, how tall I'm not. You know, everything about me is determined by some basic hydrogen bonding. And you're not going to be tested on this, but I do like my students to know something. These histones here, histones don't actually look like that. Histones are not these silver buttons or these hockey pucks that are in a bunch of pictures. Every image I see of histones, they all look pretty similar, like the little metal silver buttons, and that's not what a histone is. A histone is a protein, and we know what proteins look like. I've shown you proteins, those tertiary structures. So this is what an actual histone looks like. This is a histone. It's a protein made up of alpha helixes here, and then here is our DNA that's wrapped around it. Right. So again, this is what a histone actually looks like. So when you're going into biology class or anything and they talk about histones again, remember that's what it looks like. Histone is a protein, and they don't actually look like these little silver buttons here. There's actually some proteins, quaternary structure that's set up there. And then our DNA is wrapped around that. Not going to be tested on that, but I do like my students to know that it's not a little silver button. All right. So as far as the replication process goes, um, um, yeah, there we go. So DNA can replicate itself. Right? DNA can replicate itself, and we start with just some complementary strands here. But let me do this on the side here to a little better image. If I repeat this, so we got A, T, A, A. Um, this isn't actually how it happens. This is a very, just understand, this is a very, very basic understanding of replication. There's a lot involved in replication, and this is just a very basic overview of how that occurs. So we do some complementary strands here. T goes to A to T, T to A, C, C, A. And I'm going to do the same thing on the other side. T, A, T, G, T. Now, we started with an original parent strand here. We did some complementary strands, and then if we break that up, well, now we get two identical daughter strands there, right? So, again, it's just kind of it's a very, very basic overview on how this occurs here. So we know that DNA can replicate itself. So we now know some differences between RNA and DNA. So DNA includes deoxyribose. DNA includes thymine. But what we may not have noticed is that DNA is also a double helix. And RNA is just a single helix. RNA uses uracil. So those are some differences between our RNA and our DNA structures. It's not shown on here, but there is actually one more difference, is that DNA tends to be a really large, huge molecule. RNA is not. RNA is short and it's smaller. It's, for one, it's smaller because it's only a single alpha helix, but it's definitely nowhere near the length of a DNA structure. So as we're looking at these, so this nucleus here has what we call, I'll probably draw them. It has these what we call nuclear pores that are on the surface here. And our chromosomes, our DNA is too big to get through those nuclear pores. But if we can make a complementary strand from a small snippet of our DNA, that is small enough to slip out and carry this information outside of the cell. So RNA is a duplicate, or serves as a template, right? So our RNA is a template of our DNA structure, and that RNA, because it's smaller, does have the ability to leave the nucleus. So we're only going to talk about 3 different types of RNA: messenger RNA, transfer RNA, and ribosomal RNA. There are more, but we're only going to talk about these specific RNA structures here. So messenger RNA is the first one, and that's what's going to carry this genetic information out. So we already know that DNA, I think biologists are funny people. They make a really big deal about what they call the central dogma, right? I think they overcomplicate it. The central dogma is very simple to understand. When you go to biology and they talk about it for 3 days, the central dogma, um, it comes down to this: DNA can make more DNA. And we've already talked about that, right? We can see DNA can replicate and make more DNA. DNA can make RNA, and RNA makes proteins, right? So, when your biology instructor goes off on a tangent for 3 hours on the central dogma, just remember that in your head. Oh, DNA makes DNA, DNA makes RNA, and RNA makes proteins, and you understand the central dogma. All right. So we've already talked about DNA replication. So as far as messenger RNA comes from, that's where we're going to consider transcription, DNA going to RNA, and we consider that transcription here. So This is a DNA strand here. I know it's a DNA strand because there's thymine and it's a double alpha helix, and I know that that's going to be a DNA strand. So what happens is this big white globule looking thing here is just an enzyme called RNA polymerase, but you don't need to know that. It's just an enzyme here that has a specific job. When we replicate DNA, we can't just open it up completely and start cutting snippets off, right? So what we have to do is we can't unravel the whole thing, because if our DNA all completely unravels, then we're going to be the white globule just sitting on the ground, right? So here, little snippets of DNA are opened at a time. And this white looking enzyme here, this polymerase, moves in this direction here. And it has one job: open it up and then close it back, like a, just like a double-sided zipper, right? So here it's going to open up a small section. We're going to make our RNA complementary RNA strand, and as it's moving forward, it's going to close it back up just as it's leaving. So it opens it back up, we create some RNA, and then it closes back up again. So this is what we consider transcription. So here, right, it opens back up, it's going to close back up over here. And then our RNA is made. And then here so we have the closed back version, and then we have what we're going to call synthesized MR, messenger RNA. Now, there is another step between these two. There's one more added step. So this is what we're going to call post transcription. When we make an RNA strand here, some of the information there isn't viable, right? When you guys get your emails, I'm sure a lot of you get spam, junk mail, right? Well, there happens to be junk mail if we consider it the same kind of concept, that there's junk mail, there's spam in that RNA strand. So there is a process before we get to post transcription, right? It's got to go through our spam filter and then take out all the junk, and then we put everything back together. So that's what happens in post transcription, is that here we have some RNA that's formed. And that's the same thing that we have, and I've already kind of cleaned this up a little bit. That's what we have here. So we have some messenger RNA made, but we're not completely finished with it. It has to go through what we call post-transcription. So DNA has what we call introns and exons. Now we only need the introns. We only need the exons, and all of the introns are cut out. Introns are cut out and removed, right? That's the junk mail. That's our spam. And then the rest of it, the exons, are spliced together. And this is our final messenger RNA. Our final messenger RNA. So in here, we have introns and exons. The introns are cut out, and we get our final messenger RNA. So the introns are cut out, the exons are spliced together, and we get a template from our DNA. Now, this RNA, so we'll just call this messenger RNA here. So this messenger RNA, with all of the bases attached to it, make what we call codons. It is a code, right? So every 3 bases, we have a codon. So here, let me write this up here. So this is going to be first base. This is the second base. This is the third. base, and then it keeps repeating. So this is first base, second base, third base, first base, second base, third base. And we just keep repeating the process. So every 3 bases, we have what we call codons, and they're codes given for specific amino acids. So we talked about building proteins. Well, this is how the proteins know, how we know what order to put these proteins in, is that our DNA makes RNA, and this RNA is a template, and it gives the order of the amino acids and how to put them together. So every 3 bases here on an RNA strand, on a messenger RNA strand, have what we call a codon, and they're calling for a specific amino acid. So let's take a look at this first one. We have AUG. And if we go down all these lists here, so let's go down this whole list here, and we got AUG. And here is AUG for methionine. Notice that some have 4, this one has 6, this one has 2, this one has 2, this one has 4. So some of these amino acids have more than one codon, right? Some have 1, some have 2, some have 4, some have 3, depending on the particular amino acid that we have here. So GUC, and here's GUC, and that stands for valine. So the order that we would put this together in is that here, this would be my N-terminus, and this would be my C-terminus, right? And here I would have my protein. So my protein made from this RNA strand, calling for a specific order of methionine, valine, serine, and proline. And we now have a primary structure for this protein made from this RNA strand here. So I'm not going to provide on the exam. I'm going to provide this for you on the exam. So this is just kind of a table here. This is a list of all the codons and all the amino acids, but I don't want you scrolling through this in order to determine which amino acid you need. Is that here we're going to use this instead. So the first one we have is AUG. The first codon we have is AUG. I'm going to write that up here. We have AUG. Then we have GUC. GUC. We have AGU. GU. CCA. And finally, UAA. UAA. All right, so let's look at, this is how we're going to use it. We look at the first base, second base, third base, first base, second base, third base, first base, second base, third base. A, U, and then we go straight across. We go straight across until we get to G, methionine. We don't go up, down. Students, look at this, and I don't know what they do, but they get to U, and then they go, oh, G, oh, must be arginine, but no, that's not, we go straight. We go straight across till we get to G. The next one is GUC. So we have methionine. Let's write this. We got 5 prime and we have methionine. The next one is GUC. GU, and I'm going to stop when I get to C, and we get valine. AGU. AGU. That is serine. CCA. CCA. Proline. And UAA. UAA. And that's a stop signal. So my protein is done, and we now have a protein here. So I suggest I'll give you this on the exam. I won't give you this on the exam. But here we can figure out what different, what primary structure we have for the protein that we're building by using this here. So that's what we consider, we have post-transcription. That's what we consider translation. So translation is how we just made that protein. But there's a lot involved in what we call translation. So here, there's 3 types of RNA that we're going to talk about. So the first one we talked about was messenger RNA. So messenger RNA is here. So messenger RNA comes in, and if we take a look at this here, actually, I don't like looking at this picture. This one's much better. So put this together here, and we see this is our messenger RNA. It's going to explain everything that's going on up here. I think this was just a little bit complicated. So we have what we call tRNA, and this is our ribosomal Rna. Now let me do a different color. Ribosomal Rna. So tRNA, messenger RNA, and ribosomal RNA each have different specific jobs. Messenger RNA is our template for the protein that we're making. Transfer RNA is this key-looking structure here, right? And the protein is not made down here. What the messenger RNA has is we have a codon on the messenger RNA, and the transfer RNA has the anticodon. So the anticodon is on the transfer RNA, and it's a complementary structure of the messenger RNA, right? Remember, C goes to U for RNA, G goes to C, U goes to A, and we have that here. C goes to G, G goes to C, U goes to A. Here's our codon, and here is the anticodon. The anticodon cannot be used here. These are for codons. Do not try and use the anticodon here. So the anticodon is on the transfer RNA. And what the transfer RNA has is it brings a specific amino acid. A specific amino acid. C, G, U. C, G, U. Arginine. So this codon is for arginine. And the transfer RNA is bringing that specific amino acid, and this amino acid is arginine. So this is the arginine codon, and this is the arginine amino acid. A, C, G. A, C, G. Threonine. So here is the codon. There's the codon for threonine, and this is the amino acid of threonine. Right. So the protein is not made down here. This protein is made outside, right, up just above it here. So what happens now, so the transfer RNA is bringing the specific amino acids that we need. The messenger RNA contains the codons for that specific protein. The transfer RNA also has the anticodon, and the ribosomal RNA. The ribosomal RNA has one job, and that's to keep these in place until that peptide bond forms. It kind of works like a scaffolding, right? We're seeing bricklayers, right? They're building bricks. They have a scaffolding. They keep everything nice and steady until they're building this nice brick wall. And ribosomal RNA works the same way. It's keeping the transfer RNA and the messenger RNA in place until we can form that peptide bond. And once that peptide bond is formed, then it just continues to move. It continues to move, brings another transfer RNA with the anticodon and another amino acid, and it'll hold it in place and form the peptide bond. Messenger RNA has another codon, transfer has the anticodon, transfer RNA also has the amino acid, ribosomal RNA holds it in place, another peptide bond is formed. And it's just that repeating process here. So differences between our messenger RNA, ribosomal RNA, transfer RNA for this section. I think that covers Unit 12. So we're now finished with all 12 units.